Thanks so much for having me. It's an honor to be here and to have the opportunity to present. Thanks for taking the time to be here as well. Fortunately, we had a nice intro from Alexander Leutner about replacements, focusing on organ replacements. That saves lives, it extends lives, and of course, all our organs, as we get old, start becoming dysfunctional. And that includes the brain. And so that's what we're focused on. We're aiming to replace damaged brain tissue with new functional brain tissue, whether that damage is caused by disease or by aging. So hopefully this is the right button. Yeah. So permanent brain damage costs society worldwide in the trillions of dollars. And in general, societies are willing to pay to remedy this. It's a big problem. And that damage to the brain can be caused by stroke, can be caused by trauma, or it can be caused by neurodegenerative diseases, for example. And these estimates don't include aging per se, which is also, there's a toll to society and people not being able to function cognitively like they used to when they were younger. And damaged tissue cannot be replaced by small molecules or biologics, and they need a different approach. Single cells can't replace damaged, single cell types can't replace damaged tissue as well, because tissue is normally composed of multiple cell types that need to be working together. It needs to be vascularized so that those tissues remain viable right after transplantation. And also, most tissues, and especially for the brain, need a particular structure to be functional. And this is very important for the brain and for the wiring of the brain. So we're addressing these current limitations. People are putting different cell types in the brain for different reasons and testing them. But in order to get functional brain tissue replacement, we're taking an approach that is not reinventing the wheel, but mimicking how the brain was normally formed during development. So the brains we have in our skulls now, no scientist or engineer made them. They developed from a relatively simple precursor tissue during fetal development. And so that's what we're doing. So we're focusing on the neocortex first. That's the largest part of the brain that we use for advanced cognitive functions. And we've done a high-resolution analysis of all the precursor cells in that fetal neocortical tissue. We've done in-depth analysis of the extracellular proteome of those cells to know what their normal environment is when they're developing. So we've identified the major structural and signaling components. And from that, we can reassemble a precursor tissue that has all the precursor cell types, that's pre-vascularized, and that has the structure that is needed to develop into a normally functioning tissue. And this is what our prototype looks like. So in the middle there again is the cartoon that I just showed you that we're assembling. And on the left is a view from the top, and on the bottom on the right is a view from the bottom. So this prototype tissue appears to have all the features that we want to develop a normally structured, novel, mature brain tissue. So on the left, on the top, you see this vascular plexus that is normally there in the developing brain, and we can generate that very reproducibly now. And so that's very important. As you heard previously, for bioprinting organs, you have to have that vasculature there for after you graft so that tissue can stay alive very quickly. And on the bottom, this is sort of a specialized point to be making, but in green are these precursor cells, and for them to give rise to a normal structured brain tissue, they have to know which way is up and which way is down. And what I'm showing you here is that that's in fact what they know. So that the green labeling there is a sort of a seal at the bottom of the tissue that tells us that they all know which way is up and which way is down. So we're starting to test our prototype tissues in mice now to show how they can perform and ultimately whether they can encode useful information to their hosts. So we have this platform. We're starting with mice. We remove a piece of the neocortex, then we put in our prototype tissues to test how well they do. And these are preliminary studies that we did with mice. We're now entirely focused on human tissue that we're testing in immune-compromised mice. So the mouse work was published previously, showing, so I won't go through all the details, but just to give you an idea of what this platform allows us to test, it can test the function of the neurons. So we have neurons, if we put the grafts in the visual cortex, that over time mature and respond to visual stimuli just the same way as normal developing visual cortical neurons do. So initially, they do not respond to light at the onset of stimulus because they're too immature. Over time, they mature and become very robustly responsive to light stimulus. But not just any light stimulus. They become tuned to particular visual stimuli like a moving bar that's in this direction and not that direction, which again is typical of developing visual cortical neurons. So that's very good sign that we're on the right track and we can optimize this and get this tissue to really work. Here's another example from this early study showing that if we include vascular endothelial cells in our graft, which we do, so they can get a jump on vascularization, and on the top left here, those are labeled genetically in green for this study. And in red next to it is all the vasculature, host and graft. So you can see that they connect together, and we even have movies where you can see circulating blood within the vessels of the graft. So that's very important, and we can do that. So this is the team, the science team doing all the work. There's Alex, who developed a lot of this work before the company even started, that we're building the company on. And then we've hired a couple of tissue engineers that have really helped develop the prototype, Wyndham and Monica. And we now, because we're doing more and more tests in vivo of the functionality of this tissue, we've hired Anna, who is a really exceptional neuroscientist who does the electrophysiology and the behavior in our preclinical models. So a little bit more about the business itself. Cell-based brain treatments are still relatively new, but they have a good success rate so far. Companies like Blue Rock, Neurona, and without a doubt, Aspen will be next. In terms of success in treating specific diseases with cell-based therapies in the brain. So for Parkinson's, Blue Rock has very good early clinical results for their phase 1, 2A trials, where they see improvement in motor function, pretty dramatic improvement in motor function. For epilepsy, Neurona is introducing in their patients' brains a particular type of neuron that keeps activity at a lower level so they don't get epileptic seizures, and they have dramatic results in the clinic as well. So these companies are great. They're paving the way for what we're doing, which is the next generation of therapies for the brain, cell-based therapies, because we're doing tissue, not just cell-based. These are all single-cell sort of single indication approaches, whereas what we're doing is a tissue level replacement, so it's independent of the indication. We do have an indication that we're going for initially, which I'll tell you about, but regardless of what is the cause of the damage to the neocortex, our approach can be used to address it. So again, it's because it's tissue level, so multiple cell types, and it's structured. So the idea is that it'll be functional. So this is sort of our initial indications. Ultimately, we do want to address cognitive decline with age, which we're all going to suffer. But before getting there, we want to first prove that our tissue works in the clinic. And we've been collaborating with neurosurgeons and vascular physicians to determine what the best indication is. And that is, the clear winner is stroke initially as a first indication because it's so common. We can very closely select the inclusion criteria for our patients and the exclusion criteria, which is very helpful. And it's still, a huge market that we would stand to take a big fraction of because there currently is no effective treatment for any type of brain damage, including stroke. The next one might be a more localized type of degeneration, like frontotemporal dementia, for example. And then, more widespread from there in terms of degeneration, including normal age-related degeneration. So that's sort of the overall plan. Where we're at in the process, we started less than a year ago, so we're actually ahead of schedule in terms of meeting our milestones because of our talented scientists. We already have a prototype, so an MVP that we're starting to test in animals. And then we've also been talking to potential collaborators in terms of manufacturing and scaling this product to make sure that we're ready, in a few years, once we've done all the behavioral tests in our preclinical models, to start approaching the FDA in discussions about doing this in the clinic. We've raised $2.5 million so far in our seed round, and we're keeping that open before we, for a little while still to raise more before we go for a Series A. And I think, that's pretty much it. Our lead investors are Apollo Health Ventures. It's been great working with them. They've been super supportive on all aspects here. But we also have another quite growing group of investors who are joining us in this mission of being able to replace and reverse permanent brain damage.